We begin with a VLDL remnant particle, also called IDL, intermediate density lipoprotein, within the space of Dicey of the liver. It has one molecule of ApoB100 and a few molecules of ApoE. The triglyceride to cholesterol ratio is approximately 1 to 1. While the majority of chylomicron remnants are cleared from circulation by the liver, only about half of the IDL particles are removed. The remaining particles get processed further and returned to circulation. When there are excessive amounts of IDL particles, for example, due to high calorie intake, even less than half get removed by hepatocytes. Removal of IDL particles by the liver depends on the number of receptors on hepatocytes. When hepatocytes are cholesterol replete, they express fewer receptors and internalize less IDL particles. Uninternalized IDL particles get processed into LDL, low-density lipoprotein, primarily by two enzymes, cholesterol ester transfer protein, also called CETP, and hepatic lipase. CETP mediates the exchange of triglycerides from IDL to HDL in exchange for cholesterol ester from HDL. During this process, all ApoE proteins are transferred back to HDL. Hepatic lipase, which is similar to lipoprotein lipase, is expressed on the surface of hepatocytes. It breaks down the triglycerides into free fatty acids and glycerol, then removes them. By action of hepatic lipase and CETP enzymes, the original IDL particle is now much smaller and denser. It is called LDL, low-density lipoprotein. It is low in triglycerides and high in cholesterol ester, having a triglyceride to cholesterol ratio of approximately 1 to 5. Only one molecule of ApoB100 is associated with the particle. The LDL particle now goes into general circulation. Since LDL particles are very small, they can go in and out of blood circulation through the fenestrations between the endothelial cells of blood vessels to reach the interstitial space of most tissues. The particles may make multiple trips in and out of blood vessels during their lifetime. LDL's concentration of cholesterol is the highest of all lipid particles in the blood plasma and is responsible for delivering cholesterol to the peripheral tissues. Tissues needing cholesterol express LDL receptors and internalize LDL particles to satisfy their cholesterol needs. Once cells have enough cholesterol, they downregulate expression of LDL receptors, reducing the uptake of LDL particles. LDL particles can also deliver cholesterol back to the liver cells through LDL receptor-mediated endocytosis if the endogenous synthesis of cholesterol in the hepatocytes is not sufficient to meet the cellular needs. When tissues in the body have enough cholesterol, LDL receptors on the cell surfaces are downregulated, resulting in the LDL particles staying in the circulation longer. This increases the chances of the LDL particles getting oxidized by reactive oxygen species while in the circulation. The oxidized LDL particles are primarily internalized by macrophages resident in the subendothelial space. They express a different class of receptors called scavenger receptors, whose expression is not regulated by cellular cholesterol levels. Gradually, as macrophages internalize more and more oxidized LDL particles, they transform into cholesterol-laden foam cells, leading to the buildup of atherosclerotic plaques.